This is going to be the least theory of the talk of all the talks, probably most applied, but there'll be some algorithmic connections nevertheless. I'm going to talk about genetic disease, and we might think of it as being not so common, but it is relatively unrare, I guess. For example, I'm red-green colorblind. It's not a big debilitating thing, but it's a minor quirk. My mother-in-law has a disease called Stargardt disease, which means that in her 30s and 40s she started going blind. A friend's son has DMD. Looks like this. He's in DPS North, about I think 11th class or something like that, but confined to a wheelchair. So about 1 in 100 has a genetically driven disease. For example, 1 in 25 is a carrier for beta thalassemia in India, which if you have the disease, if not necessarily carrier, but if you have the disease, then you have to get lifelong transfusions. So it's quite a serious disease. And then another form of genetically driven disease is 1 in a few will get cancer in one's lifetime at some point, which is an acquired genetic disease, so to say. I mean, you're not necessarily born with something in the DNA, but you might acquire it over your lifetime. Okay. And if you look at the nature of genetic disease around, it falls into all of these categories, and a lot of neurological disorders, metabolic disorders, skeletal, cardiac, in that distribution. So there's a fair amount of it around. And when you have such a genetic disorder, the challenge that arises is that using conventional clinical methods, it's not easy to diagnose what's going on. There are lots and lots of instances of people going from doctor to doctor for years that end before eventually the only way to diagnose is to do sequence the DNA and figure out what might have gone wrong. And there's a very classic case that's often talked about from the University of Wisconsin about almost now 9 years or 8 years ago. And it was published in this book called 1 in a Billion, where there was a little boy with inflammation in his intestines. The cause was unclear. He went through 100 surgeries. I mean, not knowing what, all they could do is every time the phenomenon occurred, they would go and snip that part of the intestine out. Then it would show up in another part of the intestine, keep going on that way. Of course, you suspect something systemic when something like this happens. So this is an immune cause suspected, and perhaps a bone marrow transplant where you just replace the entire composition, genetic composition of the blood and therefore the immune system could be a potential cure, but a risky one. One had to be sure. And so that's when they sequenced the DNA and figured out that there was a mutation in a certain gene which plays an important role in the immune system, and that told them that that indeed replacing the bone marrow would actually do the trick. And then that worked, and the boy is perfectly fine since then and doing really well. And so this is sort of the classic case that's quoted from almost 9 years ago or so. There's a more glamorous example that a lot of you might have seen. That women have a baseline breast and ovarian cancer risk of roughly 1 in 8 and 1 in 70 lifetime risk. But if you have mutations in certain genes, that risk can go up 5, 10, 20, 30 fold. And again, there are ways to manage that risk if you are indeed carrier of that mutation, and Angelina Jolie's case sort of brought that into public limelight. Okay, so with that background, I guess the goal that I've been personally involved in, and we at Strand have been looking at for the last 5 years, has been diagnosing genomic disease. Presumably the cause, so we're looking at diseases where the cause lies in the DNA, and how do we get to the DNA and figure out what the actual cause was. The genome has about 3 billion characters, and every cell in our body, or most cells in our body, not all, but most cells in our body have DNA, 2 copies of the DNA, 1 paternal, 1 maternal, so 3 billion into 2. And a lot of these characters vary from one person to another, and that's what makes us all different. It doesn't cause a major effect as far as within quotes normalcy is concerned. But a few of these characters may be less sparing, and a change in some of these characters could have much more serious consequences. And the question we are asking is, given a patient, how do we figure out what character amongst these 3 billion into 2, so 6 billion characters, which one or many characters amongst these 3 to 6 billion are causative of the condition that a patient might walk in with. Now, to just set the context a little bit, not all so-called traits are easy to, this question cannot be easily answered for every trait that we can think of. For example, human height, it's a trait in the sense that by a definition of trait, as it's sort of not very sharply characterized by a single locus in the genome or a handful of loci. In fact, the best predictors for height today take 20,000 genomic loci and then combine them into a predictor, and yet achieve only about 0.65 correlation coefficient actual versus predicted. So not all traits are highly predictable or predictable from the genome, at least based on current understanding is concerned. But there is a subset of phenotypes, what we call Mendelian disease, which is typically caused by a single gene. Sometimes it's caused by a couple of genes or a small set, nevertheless, which typically means one or two causative characters in the DNA. And so you can localize the cause very sharply, and therefore you have some chance of identifying it. Otherwise, if it's multifactorial spread over large portions of the genome, it's very hard to take a single patient and say something meaningful. So, which typically means one or two causative characters that the patient has, but most others don't have. And it's may not be characters, may not be one or two characters, maybe one or two causative events, and I'll talk about what that means. Larger events, perhaps, which comprise many characters, but nevertheless constitute one or two events in all. And there are about 5,000 such disorders that are known, which can be localized to a single gene. Then there's a whole bunch of other things that are not so sharply localizable and which are much harder. Most of the age-related diseases that we know of, diabetes, etc., are more complex, and so I'm not going to venture into that, but only look at these about 5,000 disorders where there's a sharper cause in the DNA that one can get to. And how do we find the right characters in our patient is the question. Of course, the approach is to sequence DNA, and in the last 10 years or so, costs have dropped quite sharply enough to be able to do that on a patient-by-patient basis. And however, the process is complex and requires a set of multidisciplinary activities, all the way from deriving the right sort of lab assays to computing to biology to medicine, and things go across all of this. And hopefully in this talk I'll give you a flavor of how things go across all of most of these, if not all. And the process takes many days, if not weeks, to run on a single case. And then, of course, if you're in a real production setting, we do hundreds of cases a month, and that's a war room setting. There's a few days in the lab, followed by lots of data, followed by a lot of sequence analysis, followed by various sorts of predictive rules and literature mining, all of that in a regulatory compliant setting, and then with anxious doctors and patients calling, because there are important decisions to be made that are time sensitive. There's decisions about abortion, etc., that are critical. And so this whole thing becomes not only complex but also fairly serious activity. And so I'll give you a glimpse into how the whole thing proceeds and what the challenges are. And so the challenge with sequencing DNA is very abstractly speaking. There are many methods and a lot of evolution happening in this space, and a talk 5 years from now may look very different, but as of today, the only way to sequence DNA is by chopping it up into lots and lots of small pieces. You cannot sequence the entire stretch, 3 billion, or even the chromosomes, which are about, let's say, on average a tenth in size to 3 billion, so 300 million. You can't take these things and sequence them in one shot. You've got to chop them into lots of small pieces. The pieces would be about 150 in length, and then you can sequence each of those pieces. Okay, so what you get when you sequence the DNA is about 1 billion so-called each of these pieces. I'm going to call that a read. So you get about 1 billion reads when you sequence the DNA, which amounts to about 100 gigabytes of data for a single person. And each read is about length 150 or thereabouts. I mean, sometimes it's 100, sometimes it's 500. Read lengths are going up to 1,000 and 10,000 nowadays as well. But for most practical purposes, it's 150. How do we put it all back together? How do we put these pieces back together? And of course, the answer is that putting these back together is a one-time challenge. Once you do it for one person, You don't need to do it for every other person because any two individuals are very similar. The differences are only 1 in 1,000 characters. And therefore, once you've done it for one person, you have a reference for the entire humanity. Then when you get your patient at hand and you get these reads from that patient, you just search for each read in the reference sequence. Of course, when you search for a read, you don't need to have an exact match. As you search for this read in the reference sequence, you should allow for some differences because after all the patient is different from the reference genome in 1 in 1,000 characters. Once you do that, once you take each read and search for it in the reference sequence and you place it at the right place, you get a picture like this where many different reads stack up at each location because to begin with you've taken many copies of the DNA. I mean that's a luxury you have because there are lots of cells, plenty of DNA. So you take many copies of the DNA, take reads, chop each of them randomly into pieces so they all get cut at different places, which is a useful property. You don't want them all to get cut at the same place. And then you search for each piece in the reference genome, they all stack up. And when they stack up, you're able to see these places where all the reads are saying there's a T while the reference genome says there's an A, which means that the patient has a different character from the reference genome, which represents a presumed healthy individual. So that's how you identify differences between the patient and the average person. And of course, you have to protect yourself from noise. There's lots of noise like this. And so you need enough statistics to ensure that you've got enough votes out here that allow you to separate signal from noise. And there are of course cases where you will not get a vote in all of these reads, you will get them in only half the reads, and that's because you have two copies of the DNA, paternal, maternal, and one of them might say something different from the other. So that's what you call a heterozygous variant, and otherwise the previous picture was what you would call a homozygous variant. So both copies versus one copy. Okay, so that's the procedure that you follow, and of course now to do this at scale at 100 gigabytes of data is challenging, and I'll just briefly speak about the algorithms involved here. So your abstract problem is that you want to find all approximate matches of a pattern of length n, roughly 115 length, in a text of length n, which is roughly 3 billion in length, allowing for a small number of edits. So k is, let's say, 5 edits, and I'll come back to that number 5. How do you do that? The simple way to do it is to break the entire reference sequence into chunks of length k each. Within each chunk of length k, do a dynamic programming edit distance algorithm. There are n by k candidate windows of length k, and each of those dynamic programs can be done in order k square time. And how is that? Well, since you're allowing only k edits, if you look at your standard dynamic programming picture, you can stick to the diagonal, because if you wander too far off the diagonal, then k becomes large. So you stick to the diagonal. So this is order k. This is still order m, and so ideally the time should be m into k, because you need to compute within this diagonal. But you can actually do it in k square by doing efficient data structures, where as long as you're sticking along one of these diagonals, you're only incurring unit time. Only when you switch a diagonal, you incur time cost. So using that, you can do it at actually k into k square time. And those data structures are standard suffix tree type data structures and least common ancestor computing, etc. Okay. So overall, this is n by k candidate windows, k square time per window. So you get n into k time, and n into k is still large, you could say, and there are ways to do that even faster. So effectively, what you're seeing is a large number of dynamic programming steps being run, and this is exhaustively pretty much every dynamic programming instance is being run. You could sparsify and say, I'm not going to run dynamic programming everywhere, only where it makes sense, and there are ways to do that. And this is a paper that Cole and I published in 2002, where we showed that you can do it in actually order n time. I mean, ignore the second term here for the moment. And the way we do that is by saying, if you have a pattern and you take substrings of that pattern, long enough substrings of that pattern, several non-overlapping long enough substrings, and then you take each of those substrings and find exact matches of these substrings in the text. These are aperiodic substrings. Aperiodic just means that they don't have repeats, right? So meaning that a periodic substring is something that has a building block that just repeats again and again. At the end, there might be a little overhang, but it just repeats again and again. An aperiodic substring is anything that is not of that type. So if you have an aperiodic substring, it can't match very often. If it matches here, then the next match has to be, a good distance away. So that property ensures that, so since you're allowing for k mismatches and you have 2 k substrings, at least k of those have to match at any place for you to launch a dynamic programming instance to completely check the entire pattern. And that sparsifies things enough. The number of candidate windows then becomes n by k square, and I was planning to go through this, but perhaps I'll skip. And then the time to verify each match becomes order k, is still order k square as before, so the overall thing becomes order n. So this is essentially what we use. We take, in a nutshell, take your pattern, take lots of non-overlapping substrings, do exact matches on them, and use that to guide where you want to run your dynamic programming. Right? That's effectively, and then all the other algorithmic proofs just show that this whole thing is order in time. So that reduces your problem to now finding exact match. So you had an edit distance problem, which you've reduced to finding exact match of a collection of so-called seeds, non-overlapping substrings within the pattern. Now at 1 billion reads and each with, let's say, even 10 seeds, you've got a lot of seeds that you have to exactly match in the reference sequence. So obviously you want to pre-process the reference sequence one time so that each exact match subproblem with a seed takes time that's proportional only to the length of the seed and not proportional to the length of the reference sequence. So reference sequence one time pre-process, then every seed takes time proportional to the length of the seed, which is a very small amount of time. Of course, you could pre-process the text with suffix trees. That's what suffix trees are meant for, but they're expensive. If you put a whole human genome into suffix tree, it will be tens of gigabytes, a little more than 50 to 60 gigabytes. And then suffix trees don't have locality of reference, so there's a lot of jumping around with pointers, etc., which makes things slow in general implementation wise. So there's been a long history from 2000 on to about 2000 and early 2015 or so. There's a long history of a large amount of work in the area that would take a suffix tree and make it succinct. By succinct, we mean that essentially you try to squeeze a tree into an array or a collection of arrays. So take information that typically resides in a tree and coerce it into a collection of arrays, and ensure that the total space that is taken is actually quite small, and I'll tell you what that is. And the most elegant of that is by Ferragina and Manzini, which is that you have this 4n, or you can even make it 2n bits. So if you look at the actual DNA sequence length, right, if it's n and each there are 4 characters, so 2n bits, let's say, so allow a little bit more, 4n bits, and the rest of it is just parameterizable by a delta. So if you have a small amount of memory in your machine, you increase your delta, you can squeeze in the suffix tree into memory. Of course, your time goes up by that amount, but this trade-off is really nice. So you can really tweak your data structure based on how much memory you have, and everything is in arrays, so all the processing has benefits from locality of reference, and things become really fast. So to do your exact seed matches, this is the data structure. You use a succinct version of the suffix tree, which is now called the FM index. Essentially, the FM index is take the reference sequence, take all of its circular shifts, and lexicographically sort all of them and take the last column. Some magical stuff, but the proofs are beautiful. I would encourage you to look at it if you haven't seen it. But that's not the topic that I want to focus on. I want to tell you something that you probably don't know already, which is, okay, so then what? Now I refer to the number k at the beginning, k equal to 5 mismatches. Well, not necessarily mismatches. We're doing edit distance computation, so you can allow for insertions and deletions as well. Indeed, there are lots of cases where there are insertions and deletions. So here is a case of a family that came to us, and the heart failed in the 20s. Very young people, but the heart stopped pumping, and they had 2 characters missing in one of their genomic copies. The other copy was fine, but that was bad enough. But there was this case that was really it flummoxed us for a while before we figured out what was happening. And so this case where the organs are shifted, so what is on the left went to the right and vice versa. This is a strange phenomenon, and in fact it sets you thinking, how does the body know left from right consistently? Every individual's left is the same, right? The stomach is always on the left, the liver is always on the right, but why is it not 50-50 or something like that? That's something to think about. But when we looked at this family, 2 kids with this phenomenon, um, they had a strange situation where so this is the kid, this is the father, and this is the mother. In the two kids, they had this T that was missing presumably from all of their reads, which suggests that in both copies of their DNA there was a missing character. Okay, so you would expect if something is missing from both copies of the DNA, then it would have you inherit one copy from the mother, one from the father. So each of the parents would have one of their copies with a missing T, and the other copy was probably fine. That's why they were okay. So in the father, indeed, that was the case. So half of the reads were missing a T, which means the father had one copy with the T. So he was a carrier of sorts, not affected in any form, but silently carrying this genomic copy with a missing T. The other copy was fine, which was so far consistent. But the mother was surprising in the sense that when we looked at all her genomic copies, there was no sign of any missing T at all there. So if the children had a missing T in both copies, one was clearly inherited from the father. Where did the other come from? Of course, it could have arisen spontaneously, but unlikely that it would happen in two kids and so on. So this was really stumped us for a while. And then, somebody came up with this hypothesis that perhaps what is happening is the following, that this is the picture. The father has one copy that's proper, and the other copy has a missing T, fine. The mother perhaps has a very large chunk of the DNA that's just missing completely. The other copy is fine, and that's what you see here. But there's one copy with a large chunk that's missing, and therefore when you tear the DNA into shreds, you get very small shreds, that 150 length shreds. There's no shred coming from here at all. There's nothing to tear there. So when you take the shreds of paper and then search for them and sort of stack them out here, everything out here is really coming from this other copy. There's nothing being contributed from this copy. So the lack of a read with a missing T is misconstrued mistakenly to be no mutation at all. In fact, there's a large chunk that's possibly missing, and the children have inherited this large chunk from here and the missing T from the father. And so this is what the children's picture looks like, and all the reads are coming from here and not from here because there's a large missing chunk. There's nothing there, and that is giving us this picture, which is mistakenly again telling us that the children have a missing T in both their copies when they probably have the missing T in only one of their copies. So this took some lateral thinking to sort of arrive upon, and then we said, okay, now if this is indeed the case, then how do you find this if there's a large chunk? Now if you go back to our dynamic programming, there's a if you look at the time complexity here, the time is a function of the k out here. If k becomes very large, if k is very small, it is order n, but if k is large, then it goes cubically with k. And obviously, so if you have a large chunk and you want to allow for that many edits, that's impossible. You wouldn't even think of allowing for 5,000 edits, as it turned out in this case. So what do you do in this case? So there, of course, the natural thing to do is to say, take every read and first do the process with 5 edits if possible, and anything that fails that, you actually split it into 2 parts. So if you imagine if this was the mother's DNA and a read were to come from that, then a part of it would come from here, the rest would come from here. The middle part just is not there. So this is what it would look like, TGA, CAT. So TGA from here, CAT from here. The middle part is just not there, so it won't show up in the reads at all. So a read would look like this. It would never match unless you allowed for 5,000 edits, which is the length of this black portion, which you don't want to allow because that's too much and it will take too long to run. So the natural thing to do is to split this read into 2, break it into 2, and then you hunt for each part separately. When you do that, in this case we got this beautiful result that we took all these unmatched reads, so to say, and split them all into 2 and then search for each of them independently. And then you saw this neat stack on both sides with a large portion in between, indicating there's a big deletion here of about 5,000 or 6,000 characters, which solved the puzzle eventually. Took about 1.5 years to get to this stage because we didn't have all the systems in place. But now this is done routinely. So the point is, if you want to find really large deletions, then you need to do this so-called split read analysis. But then where do you split becomes the question. Where do you decide where to split? You don't know where to split, and so then you run the dynamic programming procedure partly and so on, and then figure out approximately where to split, etc. So those are again various pieces of computation that need to go in. overall, so when we put all of that together, and today when we look at what we do in production, about 7% of everything that we find which is causative of a problem in a patient is one of these large things, large changes, gene level, whole gene level changes, or even multi-gene level changes. 93% are small changes that change only one character or a few characters. Yeah. So then, of course, we had the task of taking all of this and implementing it in an efficient way. And what I talked about was these two pieces, which is the BWT or the FM index search followed by the dynamic programming. And then there's also the split read alignment out here. And then in between there is a sorting step because even though you search for every read, you've got to order all the reads to do some further processing. And that turns out to be painful because you can't fit it into memory, so you have to write it out to disk, and that becomes the time limiting factor in all of this. You can do all the computation that you want, writing and reading from the disk really kills, and it takes about, this was 3 or 4 years ago, 7 hours, now it's down to about 2-3 hours based on modern GPUs that you can actually put all of this into. And there are people who have taken it even further and used special programming paradigms like FPGAs and hardware paradigms to actually do it in half an hour and things like that. So all of this is doable, but now let's coming to the real problem that we grapple with on a day-to-day basis. So at the end of this process, what do you get? You compare the patient's DNA with a reference and you identify differences and you say, okay, here is a list of differences. You still haven't got the answer yet. The differences end up being 4 million in the entire DNA, even if you restrict yourself to so-called interesting regions of the DNA. An interesting region, by the way, is, so there are genes, the genome is broken into genes which carry recipes, and everything else which presumably helps decide when to invoke a recipe and when not to invoke a recipe, but doesn't directly contribute to a recipe. So for the most part, we understand the recipes for creation of proteins the best, and we understand the other parts that are controlling the creation of recipes the least. So we end up looking at just the recipes, and those are called the exonic regions of the DNA. So each gene is broken into many different exons, they are not contiguous. So if you just look at the exons which carry recipes for the creation of proteins, you still have about 100,000 variants sitting inside these exons. And the question that you grapple with on a daily basis today is, which one or two of these 100,000 is causative of the problem that the patient has walked in with. You can do some elimination quickly, well, not quickly, but through some effort. For example, you could eliminate everything that occurs in many normal people. So then you have to create databases of lots of normals. So lots of normal people have to get their DNA sequenced, and all of that has to get aggregated, and then you can look up and say, in this database there's many people with this particular variant, and so you can ignore those. The other thing you can do is to eliminate those that are in genes that are not associated with the patient's phenotype. The patient walks in with an eye, let's say a Stargardt disease, which is a condition in the retina. You don't need to look at something that causes a cardiac condition, or though there could be developmental genes that are common, very, very early in development, that are common to a lot of things, etc. But by and large, you could use the phenotype to actually eliminate. But this means a lot of literature reading and understanding what's a lot of literature curation. So that requires a whole set of tools that you have to build to mine literature fast. And the fact that a lot of literature is behind paywalls just doesn't help this process. But nevertheless, you can come down to a few thousand through this process. And then the task begins of really predicting what these few thousand would do. And then there are things that are easy to predict, and there are things which are tough to predict. And I'll give you a few examples of all of these. So things that are easy to predict. Here's a character, C becomes a T, and that causes the corresponding amino acid. This is the triplet code I'm sure all of you are familiar with. Groups of threes are what form a triplet. That triplet codes for an amino acid. Amino acids are strung together to form proteins. So if a C becomes a T here, then the change is very dramatic. An amino acid becomes an X. X is a short form for a stop instruction, which means that the program just terminates here. So if you have such a drastic change, it's very easy to predict that something drastic is going to happen. Right? So this is a fully predictable, the effect is fully predictable or largely predictable. Something like this is much harder. There's a G that's become an A, and that causes one amino acid to be replaced by another. These amino acids may have different properties, but you don't quite know whether that change will indeed cause a real problem or not. And the only way out there is to look at all the experimental evidence that's been established in literature. People often do experiments with these sorts of changes in animal systems and in vitro systems, and then publish the results of these assays. And some of those results are reliable, some are not, and you've got to sift through all of that and figure out whether this is indeed a problem. So we have an army of people who are just doing this on a day-to-day basis to serve these patients, right? So it's a lot of literature work, again, a lot of literature mining to whatever extent automated. And this is a true example. As I said, this was the case of a patient with Stargardt disease who had these two mutations. There's a lot of machine learning that is happening to in these situations where we say, can we use machine learning, take examples and then use that to learn and predict what might happen. Results are largely poor at the point, not useful in practice. Predictive capability is very poor, so stuff to be done there. There is instances where an A becomes a G, and that happens at the very beginning of the recipe. So this is called the start codon. Just like there's a stop, there's a start. And when a start changes, again, how do you predict whether the entire program comes to a halt because the start is just not there, or does something further down into the gene pick up as a new start? And there you have to discover patterns of what does a new start look like. A start should always have ATG, but it should have a motif around it, a particular pattern around it. And does such a pattern occur further down in the gene that serves as a new start? And so you look at all the other further down ATGs and say, what does the motif around that look like? And say, okay, maybe this one looks like it matches the motif well enough that it might serve as the new start. And then you've got to make a prediction on what might happen to the gene as a result of this new start. Will the gene recover its function or not? Okay. So that's the sort of things we grapple with. Then there are, I talked about largely exonic variants sitting inside the exons, but there are also variants sitting inside the introns. The introns do not contribute to a recipe, they contribute to something else, and we'll see what they contribute to. And so they become harder to predict what might happen. So for example, here inside the intron here, there was a T that became a G in this patient with thalassemia. Thalassemia is a very small gene, so if you don't find anything in the exons, there's something happening in the introns. Otherwise, it's usually it's the easiest of the diseases to diagnose and get to a solution to. So since we found nothing in the exons and there was this T to G, that's what got us looking at what was really going on. And there you've got to look at what is the role of an intron. How do our cells know how to skip across an entire intron and go from this exon to this exon while interpreting the recipe? So it turns out at the end of an exon there's a GT, and at the beginning of the next exon there's an AG. So you jump from GT to AG, and so the whole thing is spliced out. But there are lots of AGs in between. How do all of these AGs, myriads of AGs, get skipped over, and how do you get to the final AG? So there it turns out that not only must you have an AG, you must have a stretch of C's and T's just before that, and then you must have an A just before that, and that entire thing comprises a motif. And if you have all of that, then the GT, from this GT you will go to this AG and continue from there. Right? And so what had happened in this case was this T had become a G, and before the AG there was a long stretch of C's and T's. It need not be exactly C's and T's, an occasional A is okay. There was an occasional A here, and so right next to that A, the T became a G, and that just introduced a new AG out here. So instead of jumping to this AG, the cell jumped to this AG and started the protein from there onwards, and that meant a longer protein, and that caused all kinds of problems. Many such examples of special cases where you've got to look at every single mutation and figure out what could it have done. And I want to conclude with the last my story of colorblindness that I mentioned. This is the classic way to test for colorblindness. Most of you would be able to spot the number here. I can't, and I'm sure there'll be at least one or two people in the audience perhaps who cannot. The cause of this turns out to be very interesting. It's not a simple mutation where one or two characters get deleted. It's not a large deletion where 5,000 characters have gotten removed. It's more complex than that. And the cause of that, and I'll give you a little bit of background and then I'll conclude. Maybe it'll take a minute more than was allotted. So the cause is that we see red and green and blue is not relevant to this picture, red-green color blindness. So the red sensor is made from this red gene, the green sensor is made from this green gene, of which for some strange reason we have two copies, but only one is used, so you can ignore the other. And it turns out interestingly that the red and green genes are very similar to each other. There's practically very little difference between these two genes, and they were created by a duplication event in evolution. One part got duplicated. There are some differences though. There are about 12, 13 differences between these two genes. And whenever you have two portions of the genome that are very similar to each other, strange things happen. So there is this notion of recombination where a parent passes their genome to a child. The parent has two copies of the DNA. It's not, they don't pass either copy to the child. They pass a mosaic of those two copies to the child. So each one is chopped up and then a mosaic gets created and then the mosaic gets passed on. So there's this cutting, chopping and pasting that's happening. So whenever you have portions of the genome that look very similar, the cutting process goes wrong. And that cutting process can create strange hybrids like this. So this is the hybrid that I have. So I have a red gene, then I have a green red. My green gene is actually half green, half red. And then I have two other genes that are not being used, both green but not being used. So, and the cause of my color blindness is that I've got red here and I've got green and red. So my green sensor is not true green, it's much closer to red, it's halfway to red. And so my ability to distinguish between red and green is far lower than most others. And you can figure that out from just the pattern of all these reads that are placed. You pile them up and then you look at the counts and all of this, and then there's a way to deconvolute them and say, okay, this is what is happening. And so all of these sorts of varying phenomena in the genome is what we grapple with on a daily basis. And in spite of all of that, we are able to diagnose only 50% of all cases that come to us, and not only us, worldwide this is the number, about 50-60%. Why are the remaining 40% cases which are clearly monogenic based on the nature of the disease, why are they not diagnosable with the DNA sequencing that we are doing today is an interesting question, open question today. Some of this is captured in this book, which is just being released by IISc Press next month. If any of you is interested, I'm happy to send you a PDF. So thank you very much.
Any questions?
Hey, yeah.
When you said diagnose, you recommend some cutoff, some threshold or something.
Diagnose just means find the mutation which is convincingly causative of the disease. That's about it.
Is there some notion of false positive?
So in this business, you can't afford false positives. So wherever you're in a gray zone, the medical guidelines put that into a special bucket called variants of unknown significance. And you've got to label them as variants of unknown significance, leave it to the clinician to decide how they want to take action based on those. So this is a very sort of regimented area. You've got guidelines on all of these things. So the next step is some diseases are manageable in various ways. Some diseases, a small fraction are curable. If you have a missing enzyme, your body is just not producing it, you could supply it externally. Some things you can't do anything about, but you can ensure your offspring don't get it by testing them in utero, sort of prenatally, and potentially aborting. You could even take it to the in vitro stage where people are now doing, people with such, if you have parents with such problems, they actually have assisted conception, test tube in vitro fertilization. And then at the embryo stage itself, you can check whether the DNA has that mutation or not before carrying it and implanting it. So those are all the things that you do.
I was wondering, what do you see the future of complex diseases?
Yeah, so that's why, I put that slide up on height right up front and said, I don't even want to, I can't even tread into that territory because it is complex. We understand little, we know that it's highly multifactorial, not only genomically multifactorial, like height is largely just genomically multifactorial, but diabetes is multifactorial, including your environmental and lifestyle and the 20, 30, 40, 50 years of lifestyle. So that's a discussion on its own. Yeah.
Case about what happened to that person that mirrored stomach and
No, so what happens to those kids is that the mirroring itself doesn't do anything. If everything is mirrored, it's okay, right? I mean, conceptually it looks okay. But what happens is not everything gets mirrored. So the heart wasn't mirrored, which means the wiring of the blood vessels into the heart was reversed. So the left ventricle, which is the powerful thing that pumps blood to the rest of the body, was now pumping blood to the lungs, and the lungs can't tolerate that force, while the right ventricle that should pump blood to the lungs was pumping it to the rest of the body, and it wasn't strong enough to pump. So that's the problem. So really it became a cardiac event challenge, And cardiac surgeries luckily are plumbing surgeries, and you can do things with those, and you can manage. Right? So,
Take your rest of the questions offline. Let's
thank you.